Hello everyone, I'm Ojasvi Saxena, volunteer in forensic fusion. Today we are discussing about the DNA and types of DNA in forensic science and how DNA helps in solving the crime scene. Along with these, we will also discuss some case studies regarding DNA. Now let's move to the first slide is the index. In this video, we cover some basics about DNA, like we first cover introduction to DNA, types of DNA in forensic science, collecting DNA evidence at a crime scene, analyzing DNA in the laboratory, application of DNA in crime scenes, and in the last, we studied some case studies. Now, start with the introduction part. In this introduction part, we cover the basics of DNA and the basic structure of DNA. First of all, what is DNA? DNA is a group of molecules that is responsible for carrying and transmitting the hereditary materials or the genetic instructions from parents to offspring. Deoxyribonucleic acid or DNA is sometimes referred to as our genetic blueprint because it stores the information necessary for passing down genetic attributes to the future generation. DNA has two primary purposes. First, to make copies of itself so cell can divide it and carry on the same information. And second, to carry instructions on how to make protein so cell can build the machinery of the life. Next is DNA structure. Nucleic acids include DNA are composed of nucleotide units that are made up of three parts. First, a nucleobase, second, a sugar, and third, a phosphate group. The nucleobase or a base imparts the variation in each nucleotide unit while the phosphate and the sugar purpose so for the backbone structure of the bone molecules. The DNA alphabet is composed of only four characters, represent the four nucleobases. A represent adenine, T represent thymine, T represent cytosine, and G represent guanine. Now, in next slide, we see the structure of DNA. DNA in first image, so I am showing you base pairing of sugar, phosphate group, and nucleotide base, which is ATCG. As seen in the base pairing image, adenine is always paired with thymine, and cytosine is always paired with guanine. Now we move to the next slide. This is the types of DNA in forensic science. First, we start with the nDNA, which is nuclear DNA. nDNA found in the nucleus of cell. Nuclear DNA contains genetic material inherited from both parents. It's unique to each individual except identical twins. Application in forensic science, nuclear DNA is used for individual identification. It is commonly analyzed through STR, short tandem repeats profile, which examines specific areas of DNA to create a genetic profile unique to the individual. It is considered highly accurate and is widely used in criminal investigation and paternity testing. Sample sources blood, saliva, semen, hair roots, skin cell, and the other bodily tissues. Now, the image of nuclear DNA. After this, we discuss about mtDNA, which is mitochondrial DNA. Mitochondrial DNA is found outside the cell nucleus in the mitochondria, unlike nuclear DNA. mtDNA is inherited only from the mother. Application in forensic science. Mitochondrial DNA is used when nuclear DNA is unavailable, often in regarded to sample. Since mitochondrial DNA is more abundant and stable, it's useful in cases involving bones, teeth, hair shaft, or other samples where nuclear DNA may not be retrievable. Sample sources, hair shaft, bones. teeth, and degraded remains. Now, move to the images of mitochondrial DNA. In human, mitochondrial DNA consists 16,569 base pairs. Now, move to the next is touch DNA. Touch DNA is the genetic material left behind from skin cells when someone touches an object. It contains only traces amount of DNA, which makes it harder to analyze. Application in forensic science. Useful in cases where an individual has touched an object, example weapon or clothing. Because it obtained in low quantities, specialized techniques are required for successful analysis. Sample sources, object or a surface that a person has touched, like weapons, clothing, or tools. Now, move to the next slide is the collecting DNA evidence at a crime. In this, we study some sources of DNA first. First sources of DNA is skin. Evidence from skin contact, also referred to as touched evidence, can be collected and used for forensic DNA analysis. One example of this evidence is shed skin cells that are found on worn clothing, which is frequently encountered in crime scene investigation. Second source of DNA evidence is hair. Hairs include scalp and pubic hairs, frequently constituent biological evidence that is found at the crime scene. The isolation of DNA from intact hair roots is routinely uh, used in nuclear DNA analysis. In comparison, the quantities of DNA in telogen hair roots are considerably less than the DNA that is found in the roots of energy hair. Now, we move to third source of DNA evidence is bone. A number of methods are used to identify human remains. For example, the identification of facial characteristics, the recognition of individualizing scars, marks, and other specialized body features, the matching of dentition with pre-mortem dental X-ray, and the comparison of fingerprints. The fourth one is teeth is the sources of DNA evidence. The characteristics of teeth, their alignment, and the overall structure of the mouth provide information for identifying a person. The use of dental records, such as X-ray and dental cards, can allow dental remains to be connected to a victim. After sources, we move to the collection of evidence. First is swabbing. Use sterile swab to collect trace DNA from sources like weapons, clothing, and other items. Swabbing should be done gently to maximize collection without damaging the sample. Cutting. Next is cutting for items like fabric with visible biological stains. A small section can be cut out and preserved directly. Next is scraping. When DNA is dried on the surface, gently scraping the area can gather particles without damaging the DNA. Next is avoid direct contact. Directly handling samples can cause DNA degradation, so only clean, disposable tools should be contact the evidence. After collection, we are moving to analyzing DNA in the laboratory. First, we start with the methods of DNA extraction. Extraction methods are various types. But in this video, we will discuss about two extraction methods. First extraction methods are phenol chloroform and the other was organic solvent. Extraction with phenol chloroform. These methods is also called organic extraction. Major steps include the following: cell lysis and protein digestion. These steps can achieve by digestion with prolytic enzymes such as proteinase case before extraction with organic solvent. Now, extraction with organic solvent. The removal of protein is carried out by extracting aqueous solution containing DNA with a mixture of phenol, chloroform, isoamyl alcohol in the ratio to 25 is to 24 is to 1. After these, we see the image. In this image, we see DNA extraction using organic solvent and the concentrating DNA solutions using filtration device. After this, we discuss DNA quantitation method, which is slot blot assay. The slot blot assay was used to detect human genomic DNA in a sample. An alkaline solution is added to the genomic DNA sample, which denatured DNA. Generating single stranded DNA is necessary for DNA to be cross-linked onto a nitrocellulose membrane. The DNA sample is then spotted using a slot blot device onto a nitrocellulose membrane. The single-stranded DNA is then immobilized onto a nylon membrane. The targeted sequence is revealed by hybridization with a labeled 40 nucleotide probe complementary to a primate-specific alpha satellite DNA sequence at the D17Z1 locus. After this, I'm showing you the image of slot plot technique, how it works, and what device we use in slot plot. After this, we discuss about the very common method of DNA extraction is polymerase chain reaction, also known as PCR. These techniques was developed in 1983 by Carey Lewis. PCR may be able to generate millions of copies of small segments of DNA. The principle of PCR. The PCR technique is based on enzymatic replication of DNA. In PCR, a short segment of DNA is amplified using primer-mediated enzyme. DNA polymerase synthesizes new strands of DNA complementary to the template DNA. The DNA polymerase can add a nucleotide to the pre-existing 3' OH group only. Therefore, a primer is required. Thus, more nucleotides are added to the 3' primer end of the DNA polymerase. Steps of PCR. The PCR involves three major cyclic reactions: denaturation, annealing, elongation of the new DNA strand. After this, I'm showing you the image of PCR technique, how it works, and how double-stranded DNA converted into single-stranded DNA and then form the new DNA strand. After this, we study the application of DNA in solving crime. First, identifying suspects. Crime scene DNA matching. When DNA is found at a crime scene, such as in blood, hair, or skin cells, forensic scientists can create a profile and compare it to suspects' case. Second, linking crime scene. Serial crimes and offender tracing. DNA profiles found at different crime scenes can be compared to establish links between them. Third is sexual assault and abuses cases. Identification. DNA collected from victim or crime scene in sexual assault cases can be compared to suspects or database profiles, helping to identify the subject. Fourth one is missing person and disaster victim identification. Identifying human remains. DNA can be used to identify human remains by comparing samples to relatives' DNA profiles. At last, we discuss some case studies. Now, we start with first a case study on Shraddha Valkar murder case 2023. Shraddha Valkar, a 27-year-old woman, was strangled and chopped brutally by her live-in partner, Aftab Punawal, and was thrown off into the Mehrauli jungle in the national capital, Delhi. He killed her on May 18, 2022, and the case came to public attention on November 11. DNA used in this case. DNA analysis of body parts found in various locations confirmed the identity of Shraddha, establishing critical evidence in the case. Outcome: this case underscores DNA's crucial role in linking dismembered remains to the victim in crime. After this case, we will discuss about Swati murder case in Tamil Nadu. The Swati case involved the murder of a young software engineer, Swati, at the railway station in Chennai on June 24, 2016. The murder was caught partially on CCTV, which led authorities to identify Ram Kumar as the main suspect. DNA evidence. DNA samples from the victim's body and the crime scene helped confirm the identity of the attacker. DNA evidence play a crucial role. Blood stain found on the Ram Kumar's clothes matched Swati's DNA, directly linking him to the crime. The forensic evidence strengthened the case by providing proof of the suspect's presence at the crime scene and involvement in the attack. After this, the third case is something we all know very well, is the Nirbhaya case 2012. On the dark and cold night of December 16, 2012, a 23-year-old woman was brutally assaulted and raped in a moving bus in South Delhi. Jyoti Singh, a 23-year-old female physiotherapy intern, was beaten, gang raped, and tortured on December 16, 2012 in a neighborhood in South Delhi. She was traveling with her friends Avindra Pratap. DNA evidence, DNA samples from the victim body at the crime scene helped confirm the identities of the attackers. Outcomes. The evidence led to the conviction and eventually execution of the perpetrators, marking a landmark in using DNA evidence to secure justice in the high-profile case. Alright, thanks for watching. And if you have any question, drop them in the comment box. Thank you so much.